Bismillahirrahmanirrahim. Uh, I'm Dr. Shafique. I am working as assistant professor in the Department of Microbiology, GCUF. So the topic today, which I'm going to present, is the bacterial contaminants of milk collected from various specimen sources. So first of all, we will distribute our talk into introduction, objectives, materials and methods, results, and conclusions. So almost 33 different microorganisms, they are responsible for various foodborne diseases such as bacteria, viruses, toxins, and different chemicals. And approximately 600 million people, including the children, which are having the age less than 5 years old, they got sick annually due to consumption of these contaminated food. And almost diarrheal diseases, they contribute more than half of the cases of the foodborne diseases. And number of diarrheal disease cases is almost 550 million, and number of deaths, they are 230,000 every year. On the onset of the diarrhea is mostly caused by the consuming of the contaminated food, such as eggs, contaminated raw or undercooked meat, dairy products, and which is due to be followed by the pathogenic organism like E.coli, Salmonella, Cumulative, and virus. According to the CDC, milk and other dairy products, they have a high nutritional value due to its proteins, lipids, and vitamins. And there are other important bioelements like calcium, magnesium, selenium, which provide strength to our bones and also reduce diseases. And milk-borne outcomes or outbreaks, they are recorded as 2 to 6% due to the bacterial contamination. And average number of outbreaks, they are linked to the raw milk that are more than 4 times higher during the 2007 to 12 compared to the 1993 to 2006, according to the CDC report. In Pakistan, up to now, few studies have reported the prevalence of pathogens in milk and milk products. And Salmonella is the major responsible agents of foodborne infections. And contamination of the milk with the Salmonella and E. coli has been reported in a study which was conducted in Peshawar and Quetta. In humans, Klebsiella is also a main source of pneumonia, septicemia, UTI, and also life-threatening septic shock. According to another studies, which was conducted in 2017, it has been reported that more than 31 different strains of Klebsiella has been found in raw milk samples of different animals. Further, the analysis of raw milk and its products, they are they are included the different microorganisms, and that's why its quality of the those milk products, and it's very important. And these protocols, they may enhance the shelf life of the milk and benefits for the consumers. Quality milk is the right of consumer. It should be free from harmful toxins and bacterial count. It should be less than 1.5 to 10 to power 5 CSU per ml. And quality of milk, it can be analyzed to different ways, to physical and chemical means, such as by determining the density of milk, fat concentration, and most importantly, microbial analysis. That's very important to determine its the pathogen microorganism, whether it's contaminated any pathogens. So objective of this study was to investigate the bacteriological quality of the raw and pasteurized milk samples that was collected from various regions of the Faisalabad. And also the isolation of the potential pathogenic bacteria from the raw and pasteurized milk. And what's the methodology which we used during this study? The collection of the specimens. Total 110 milk samples were collected. Among these 110 samples, 100 samples were collected from raw milk and 10 samples were of pasteurized milk. Out of 100 raw samples, 25 samples were collected from dairy farm milk and 25 samples from milkmen, and 50 samples were collected from the milk from different milk shops. And we also want to investigate whether the pasteurized milk of different brands, whether they have any pathogenic microorganism. So we also collected 10 samples from different milk pair of different brands. So bacteriological analysis. In this case, we use methylene blue reduction test, BOT. In this, we determine the quality of milk, and we determine the reduction time. And if the reduction time is 5 hours and above, it gives a very good quality milk. And if the reduction time is 3 to 4 hours, it's good quality. And 1 to 2 hours reduction time means milk having a fair quality. And if the reduction time is less than half an hour, then the quality of milk is very poor. And bacteriology analysis, second, is the standard plate method, in which we make different tenfold dilutions, single dilutions of the samples, and then we perform the spread plate methods. Third is the total coliform counts. So in this case, we also determine the most probable number method, ambient method, and further we perform different tests, presumptive tests, confirmed tests, and complete tests. And finally, we those samples we also further confirm on by using different gram stainings and biochemical tests. Now, results. So, bacteriological analysis, in case of bacterial analysis, the results of the methylene blue reduction BRT test showed in this figure, you can see in tube 1 and tube 3 to 10, the dye has reduced completely, while in tube number 2, the dye is not reduced completely. And if we see in this graph, you can see almost 88 samples having poor quality, 12 have fair quality, and on the pasteurized milk, 10 samples they are of a good quality milk. Standard count plate method. In this case, we determine the total bacterial count of the dairy farm milk samples from milkman milk samples and milk shop milk samples. So we found that on the dairy farm milk sample, it was 625 CFU per ml, and milkman sample is 7.5 into 10 to 5 CFU, and milk shop sample, it was found that the total bacterial count was 7.6 into 10 to 5 CFU per ml. Then these were cultured on the different agar plates, colonies were nutrient agar plates, and we found that there was significant correlation was found among the dairy farm milk samples and the milk shop milk samples. Presumptive test was performed, and we found that 17% of the raw milk samples they gave coliform unit more than 110 per ml, and the pasteurized milk showed less than 7 per ml. So in this case, only the 70% of the raw milk sample they showed coliform. unit more than 100. So it should be the normal value is less than 100. It should be less than 100 per ml. Confirm test. Then these coliform, different coliforms, they were also we confirmed on different agar. EMB agar, we for the termination of the E. coli, we cultured on the EMB agar, and total we found that 46 coliform were confirmed in case of the raw milk, and only two in two samples we in case of pasteurized milk we found E. coli in those samples. And further we did confirm by performing the different biochemical tests. In case of we found that indoor test and methylene red test they were found positive, while Bogus Prosecutor and separate tests they were found negative. Further, we determined the distribution of Klebsiella in these specimens, and we determined the growth of Klebsiella on monkey agar, and we found that almost 9 samples they were found for the positive for the Klebsiella, 7 samples in milk sample, and 6 were of dairy farm milk samples. Only 1 sample were we determined and the Klebsiella in only 1 sample of the pasteurized milk. And similarly, we determined the biochemical test for the Klebsiella. It showed that indoor test and methyl red test they were negative, and the Bogus Prosecutor test and Bogus secret test they were found positive. And then we determined the distribution of Salmonella samples on the SS agar, and we found that 8 samples were in 8 samples the Salmonella were found, and pasteurized in pasteurized sample only 1 sample this Salmonella was detected. And biochemical test showed that indoor test and Bogus Prosecutor test was and Cypress test was negative, while methyl red test was positive. Cumulative results showed that in case of raw milk, among the 100 raw milk samples, we found E. coli in 46 samples, Klebsiella in 22 samples, and Salmonella in 15 samples. While in case of pasteurized milk, out of 10 samples, E. coli was found in 2, Klebsiella and Salmonella in 1 samples. Okay, finally, we can conclude that the majority of the sample, almost 80%, they were found of poor quality, showing TBC of 1.6 million CFU per ml, and the presence of pathogenic bacteria such as E. coli 43.6%, Klebsiella 20.9%, and Salmonella 14.5% in milk samples of that of great health risk for the human. So overall microbial quality of raw milk transported in the festival, it was found inferior. So recommendation, and in order to for proper hygienic measures should be adopted to minimize the chances of contamination of the milk, and disease animals should be isolated, and pasteurization of the milk that is necessary before processing of the milk. Thank you.